We offer innovative approaches to a range of molecular interaction problems. We're constantly looking for new ways to refine our techniques and to provide investigators with new approaches to solve their problems. I came here in 1983. My background is in chemistry, in particular in physical chemistry and biophysical chemistry, and my entire career has been spent looking at problems in molecular recognition. Our focus is on enabling investigators to measure interactions between molecules that are important for human health and disease. And we offer a range of biotechnologies that enable people to figure out how quickly, how tightly, and with what specificity two molecules, let's say two proteins, or a protein and a piece of DNA, or a protein and a lipid interact with each other. And our technologies are based on light. So we offer, for example, surface plasmon resonance, an optical technique that measures in real time how quickly and how tightly, let's say, a protein binds to its ligand or a drug binds to its target. From that information, we can also determine selectivity. So this is an important technology used in screening new drugs. We also use circular dichroic spectroscopy as a measure of the conformation of a molecule. So oftentimes a molecule can exist in, let's say, a resting or inactive conformation, and then a drug can bind to it and change its conformation to an active form. And so we can measure information about the structure of a molecule with a technique that we call CD. Our work has primarily been with investigators here at the Medical Center, but we also have collaborations with colleagues at UNC Chapel Hill and the University of Maryland, and more distantly, colleagues in Europe and Holland, the Netherlands and Italy. And we are especially interested in partnering with the pharmaceutical industry for drug discovery. The kind of projects that we do require a lot of interactions with the investigator. So the first thing we do is send the investigator a questionnaire and say, what's the biological problem that you want to answer? And then we sit down and say, of the technologies that we have available, which one of those is most appropriate to your problem? Then we design a pilot experiment to determine feasibility. That takes about one day. And then we analyze the data and sit down with the investigator and figure out the next steps, which usually leads to a more complicated series of experiments requiring a lot of back and forth analysis and then redesign of the experiments.